Okay. So finally, the spermatozoa reach the egg. It senses a different temperature, is swimming very fast and strongly, and it senses the change in concentration of chemical and molecule messages from the cumulus, and finally it reaches the egg, high in the ampulla. Now, the first layer of the egg that the sperm encounter is obviously the cumulus, and the sperm need to be able to swim through the cumulus and reach the zona pellucida. So in the area of cumulus and near the zona pellucida, we have what is called the acrosomal reaction. So the membrane of the surface of the head of the sperm fuse with the membrane of the acrosomal vesicle to release the content of the acrosome in the cumulus and toward the zona pellucida. This is important because the content of the acrosome include enzyme that allow the digest the cumulus, and allow the sperm to swim toward the zona pellucida. Also, the acrosomal reaction allow the exposure of some of the proteins that require, some of the receptors that are required to the recognition of the sperm and the zona pellucida proteins. So they have some receptor that are exposed after capacitation that bind the zona pellucida protein, in particular the zona pellucida 2 and 3, and allow the sperm to move from the cumulus through the zona pellucida and allow the sperm to reach the membrane of the egg. Now here, in the membrane of the egg, an important protein that we mentioned before is needed, which is izumo. Izumo, after acrosomal reaction, is now exposed on the surface of the membrane of the sperm, and izumo bind two proteins, Juno and CD9, that are expressed on the membrane of the egg. The binding of Izuno and Juno and CD9 are fundamental for the fusion of the membrane of the sperm head and the membrane of the egg. After the fusion of this membrane, the nucleus containing the paternal DNA is released inside the egg, and we have nuclear fusion, we have fertilization. So here again, another sketch representing the moment of fusion of the membranes between the sperm and the egg, and the movement of the nucleus containing the DNA or the paternal DNA inside the egg. Immediately after fertilization, so immediately after the membranes of sperms and egg are fused, and the nucleus of the paternal nucleus is entering the egg, we have what is called another block of polyspermy. So how this other block of polyspermy works? This is called the slow block of polyspermy. Immediately after immediately after the fusion of the membranes of the sperm and the egg, a layer of cytoplasm that is near the membrane of the egg release what are called the cortical granule content. So there are cortical granules that live just below the membrane of the egg, inside, of course, the egg, the cytoplasm of the egg, and release their content immediately after one sperm fuse with the membrane of the egg. These cortical granules contain enzymes that digest the zona pellucida. In particular, they digest the zona pellucida proteins 2 and release zona pellucida protein 2 from the zona pellucida matrix. In this way, the zona pellucida proteins, the zona pellucida matrix, detach from the membrane of the egg. So it really detached from it. So any other sperms that is able to bind to the zona pellucida cannot reach the membrane of the egg. So here what we see in this beautiful picture is a mature and fertilized egg. You can see that this mature and fertilized egg is is a secondary oocyte. We can see here the chromosomes, so the DNA of this egg, which are organized on a spindle that is pushing toward one side. You can see this bulb there that represent a forming polar body. So we have a secondary egg at the beginning of meiosis 2. Immediately after fusion of the membrane of the sperm and the egg, we can recognize here the point of entry of the DNA of the sperm. So here in blue, this blue here represent on the left-hand side represent the point of entry of the sperm, and you can see this green circular structure that is called an aster that is formed from the centriole of the sperm, while on the top of the picture you can see two DNAs, again in blue. One represents a secondary polar body, while the other one internal of the cytoplasm represent the maternal nuclei. So you can see that only after fertilization, this egg finish meiosis. In fact, only after fertilization, we have the final step of meiosis with the formation of the extra polar body. Now, this aster, the job of this aster that is formed from the point of entry of the male nucleus, the job of this aster is to put in contact the male and the female nuclei. So when the two male and female nuclei are fusing together, what they do, they organize their chromosomes in a common spindle, which is formed starting from the centriole that comes with the sperm. And one fun fact is that although after the fusion of the male and female nuclei, and after the forming of the chromosomes, male and female chromosomes in the same spindle, although they do all this, what they don't do is to form a nuclear membrane around them. So in fact, the first cells that have a nuclear membrane, so they have a real diploid nucleus, are the cells that are formed after the first zygotic division. So they are when we have an embryo with two cells. So what did we learn now? So in this lesson, we should be able now to recall the structure of sperm and explain the significance of its formation, recall the structure of the ova, the eggs, and explain how it is formed, describe the difference between meiosis of the two gametes, describe, of course, the maturation steps for the sperms, and describe the process of fertilization.